tend to talk with my hands, so I'm going to apologize in advance if you can't hear me because I'm gesturing too much. But thank you. I'd like to echo the other speakers. Thank you to the organizers for the kind invitation and really congratulate them on an extremely successful meeting so far. So I study zinc, and I've done a lot of work in zinc relative to its role as an antioxidant, its role in protecting against oxidant stress and genetic damage. And I'm not going to talk to you at all about that today. And I'm going to share with you some more recent work that we've done in the lab that really is trying to establish effects of zinc more on epigenetic alterations rather than genetic alterations, its role in inflammatory processes, especially in an aging population. So just as a general background, so I've been very interested in zinc for quite some time, largely because it is involved in so many diverse processes. So there are over 300 zinc-containing metalloenzymes and well over 1,000 zinc-associated proteins that use, for example, zinc to help bind to aiding transcription factors to bind to DNA. So I always kind of tell my students, if you're ever on Jeopardy or in Trivial Pursuit, and your category is metal-containing protein, which really never happens, otherwise I would win a lot more Trivial Pursuit games. But if you are asked that question, to guess zinc, because chances are it'll be zinc, because it, again, is a cofactor in so many different proteins and complexes. And because it's a part of so many different categories of proteins, its function is also very diverse. So basically, if you are a cell and you need to divide, then you need zinc. If you are pregnant, you require zinc. So zinc deficiency also is categorized as a teratogen. So if you're pregnant and have coinciding zinc deficiency, the chances of you having a birth defect is much higher. Outside that, again, I've really focused on zinc as an antioxidant function. It's a cofactor in antioxidant enzymes like copper zinc superoxide dismutase. It's a factor in several DNA repair proteins. So again, I've really focused a lot of my work around maintaining DNA integrity and cancer risk. Outside that, though, zinc also plays an important role in immune function, and I'm going to focus mostly on that for the rest of my talk. The other important thing to realize about zinc, that in contrast to some of the other trace elements that are essential, such as iron, there isn't a large long-term storage form of zinc. So unlike iron, where we have ferritin and hemosiderin, most of our zinc in our body is bound to functional proteins rather than stores. So if you don't consume enough zinc or have a condition that you secrete or lose a lot of zinc, the capacity of your body to combat that is fairly limited. And what is going to be as a consequence is functional changes, because again, you don't really have a high capacity to be able to buffer changes in intracellular zinc. So again, zinc is really also very important in the normal development and the function of an immune system. So another consequence you see as a result of zinc deficiency are changes in both the adaptive and the innate immune systems. And I'm going to go over some of these processes in a little bit. So where do we get our zinc? So for a healthy adult male, healthy adult female, you need about 11 milligrams if you're a male, 8 milligrams if you're a female. And in contrast to usually the types of nutrients that you think about, or foods you think about for antioxidant-rich foods, zinc is not found in green leafy fruits or vegetables, which is what you usually associate with antioxidant-rich foods. Zinc follows protein, so it tends to be a lot of protein-rich foods. So here in Paris, one of the first days that I was here, one of my colleagues found this restaurant, Le Petit Zinc, for me. So I had to eat there, and of course, what was highly prominent on their menu were oysters. So oysters are one of the #1 zinc-containing foods. So these couple of oysters provides you well over the RDA for zinc. So if you want to make sure you're getting enough zinc, head over to the Petit Zinc and have a couple of oysters. But other foods that contain zinc, again, tend to be protein-rich. So lean red meats, nuts and legumes, and other protein-rich plant-based products also have quite a bit of zinc. But the limitation with some of these plant-based products is that they also contain phytates or inositol triphosphates. So the bioavailability of this zinc is also less because the phytates bind up the zinc in the gut and make it less bioavailable. In human zinc nutrition, one of the banes of our existence is there currently is not a sensitive and specific biomarker for zinc deficiency. So if you suspect that you're not getting enough zinc, and we've clearly been able to show in a lot of preclinical models and clinical models that if you don't get enough zinc, there are very serious consequences in terms of cellular function and health outcomes. But we do not have a good test for you to take. So what's currently used in the clinic are plasma zinc levels. And again, it's really not a very good marker in terms of either being sensitive or specific for zinc deficiency. And I'll show you a little bit of data to illustrate this point. So quite some years ago, we did a clinical study in collaboration with Janet King and Ken Brown at UC Davis and UC Berkeley, where we took subjects, brought them in, and fed them a very controlled diet and manipulated their zinc status. So what this graph shows is the design of this study. And so these were healthy adult males. So these are not older individuals. These are healthy adult males, all under the age of 50. We brought them into the study center, and for 2 weeks, we gave them a zinc adequate diet. Again, reminding 11 milligrams is an adequate zinc level to ensure that they all had adequate zinc status. And then after this 2-week period, what we did was put them on a zinc depletion diet. So the first week, these guys got 0.6 milligrams of zinc per day, so less than a milligram of zinc a day, plus a high phytate diet for 1 week. This was not a food-based diet either. This was a liquid diet to ensure that we could quickly deplete these subjects. And then for an additional time, so this is about another 6 weeks, we put them on regular food diet that contained a low amount of zinc, 4 milligrams of zinc per day. So again, adequate period, a zinc depletion period. And then at the end of the depletion period, what we did was supplement the subjects with 20 milligrams of zinc for 1 week, and then also fed them a zinc adequate diet. So again, we have an adequate period, a depletion period, and then a repletion period. And we got blood samples and urine samples from all these studies. And I'm not going to go over a lot of the data that we found, but the one thing that I want to point out, again, illustrating that plasma zinc levels are a terrible biomarker. So this are the plasma zinc levels in each of these subjects. So again, reminding here is the beginning of the study period. This is the zinc adequate a diet period. This is halfway through the zinc depletion period, and this is at the end of the zinc depletion period. And note there is no change in the plasma zinc levels, which clearly shows really not, these poor guys, again, had to eat a liquid diet, low zinc for an entire week, and then an additional 6 weeks of low zinc. And our current marker for zinc status doesn't change at all. A little bit of increase with the zinc supplementation, but again, I show this mainly to, again, illustrate the fact that zinc status is very difficult to assess and really is problematic when we look at some of the clinical trials using, for example, zinc supplementation. The effects of repletion versus supplementing someone who's already adequate is going to have very different effects, and I think largely accounts for a lot of the inconsistencies we see with some of the clinical trials with zinc. So who are the people that are susceptible to zinc deficiency? So on the first day of the conference, one of the micronutrients that came up in terms of worldwide issues for micronutrients was zinc. So worldwide, zinc deficiency is a major problem. It's one of the more prevalent micronutrient deficiencies worldwide. But even a lot of the zinc research has focused on developing countries, but even in developed countries like the US, zinc is also a major issue. So in the United States, about 12% of the population overall does not consume the EAR, the estimated average requirement. So this is, as Dr. Zeisel had mentioned in that session, is the average amount that zinc is required. So this is below the RDA. So if I crunch the numbers for the number of subjects getting the RDA, it would even be higher. But what's interesting is if you're over age 55, that number markedly increases. So if you're over age 50, 40 to 45% of the population are not consuming adequate zinc. And on top of that, when we look at an older population, even if they are consuming enough zinc, that they don't appear to utilize it as properly. So there's been several studies that have shown, at least in supplementation, if you supplement an older individual and match them to a healthy control, their plasma zinc levels do not change or do not elevate. So a young person, you give them a high zinc supplement, their plasma zinc levels elevate quite markedly. An older individual, we don't see that same increase in plasma zinc levels. So their utilization of zinc appears to be compromised. So again, this is kind of a double whammy in terms of zinc status, where an older population both appears to consume less dietary zinc, but even if they are consuming zinc, they may utilize it differently. So this is certainly a population that is going to be more at risk for the health consequences of zinc deficiency. So one thing that we see with aging is a lot of immune decline. So I want to talk a little bit about immune decline and then link it back to zinc status. So aging is associated with several types of immune defects, both in, again, the innate and the adaptive system. And it's a situation where you both get compromised immune system and also coincides with exaggerated immune system. So what happens as we age is there tends to be an involution of the thymus, which causes an inability to produce several key immune cells, in particular both the T cells and the B cells, which are responsible both for adaptive and innate immunity. But this, in the end, ultimately causes a person to be more susceptible to infectious disease and vaccine efficacy, which is quite well known in terms of older individuals being more susceptible to infections and that type of thing. We also know just from the population data that this is true, that as we get older, again, susceptibility to respiratory infections and other infections are more seen as we age. And again, it's also well known that a vaccine efficacy is also reduced with age. But then again, at the same time, although there appears to be with age this blunted immune system, there is also at the same time an overactive immune system relative to inflammation. So with age, there's also this concept of inflammaging, where aging is also associated with an increase in chronic inflammation. And it's thought that this chronic inflammation can lead to several chronic disease states and also a lot of the pathology that's associated with aging. So what's interesting, though, if I go back and insert zinc deficiency for aging, that I could replicate each of these slides in terms of the phenotype that you see with zinc deficiency. So the same thing, when you become zinc deficient, you see this similar atrophy of the thymus, you see increased susceptibility to infections, you see decreases in immune response, and you also see an increase in chronic inflammation. And especially these pathways related to chronic inflammation with alterations in zinc status has been a major focus in a lot of the research that we've been doing. So again, it's interesting that a lot of the immune defects that we see with age are very much mirrored both in animal models and people when they become zinc deficient. So again, what happens when you become zinc deficient are a lot of different things, but with respect to the immune system, again, you have a reduced thymic output, relative to this involution of the thymus. You also have lymphocytes are decreased, depressed adaptive immunity due to decreased B cells, and impaired host defense. At the same time, again, with zinc deficiency, similar to with aging, you also have this chronic inflammatory state that also appears to be perturbed when you have low zinc. So there's been a lot of work on both sides in terms of zinc deficiency, increasing immune response, but also inflammatory diseases appear to affect zinc status. So, for example, during sepsis, there is a rapid change in zinc status, especially in local organs such as the lung. During allergy, inflammatory processes as well, that you see a movement and redistribution of zinc as well. And then increases in inflammatory states and zinc deficiency also can induce oxidant stress and DNA damage that also can promote inflammation. So we see this kind of feed-forward system where zinc inflammation are feeding forward the process to promote more chronic inflammation. And again, we've been very interested with understanding what happens specifically during age, because again, this is the population that is most highly susceptible to zinc deficiency and also susceptible to these inflammatory and immune defects. So we wanted to ask the question, does age-related zinc deficiency contribute to immune dysfunction and chronic inflammation during the aging process? So some of the first things that we did was just to look at zinc status in this case, animals that are either young or old. So this is simply looking at zinc levels in either a 2-month-old mouse versus a 22-month-old mouse. And we've several talks before this have talked about using these aging models, but you can see here, here we have plasma zinc levels in the young and versus the old animal. Both of these animals are fed a zinc adequate diet. So for a mouse, this is between 30 and 50 parts per million zinc level. So they're not fed, they're fed the exact same zinc adequate diet, yet the older animal has a markedly lower level of zinc, even though they're eating enough zinc. If I compare this to a young animal that I've given a low zinc diet, less than 1 part per million, the plasma zinc levels are fairly equivalent. So basically this old animal, even though they're eating adequate zinc, now looks like a zinc deficient animal. When I look more closely at various organs, so this is flow zinc 3, which is a fluorescent marker for zinc status. So it's largely looking at intracellular zinc levels. Here we have the thymus, the spleen, and bone marrow. So these all are organs that are responsible for deriving a lot of these immune cells. We again see this loss of zinc in the older animals, specifically in these organs. Interestingly, if I look at other organs, such as the liver and the lung, I do not see these changes in intracellular zinc levels. So the loss in intracellular zinc does appear to be fairly specific to some of these immune cells. What this last bar is, these are bone marrow-derived dendritic cells. So what we do is take the cells from the bone marrow, and then we need to culture them ex vivo and differentiate them into dendritic cells. So dendritic cells up here are largely responsible for antigen-presenting function. When they encounter inflammatory stimuli, such as TNF or LPS, these dendritic cells mature and activate, and they are very potent in terms of eliciting inflammatory responses. The reason why I want to point this out more specifically is we have to culture these cells again for 7 days, and we culture them in a zinc adequate media. So even once we take the cells out and culture them in a zinc adequate media, they still have this loss of zinc. So this aging process is causing some specific alterations in these immune-derived organs that's compromising zinc status. To try to understand this, so there's 2 things we're trying to understand. Why does the older animal have this increased susceptibility to zinc deficiency? And how does this contribute to inflammatory processes? And there's certainly a lot of increasing evidence that suggests that inflammatory processes have, and dysregulation of epigenetics, not just genetic alterations, contribute to some of these processes. And I know this afternoon we're going to hear a lot more in terms of epigenetics and aging, so I'm not going to spend a lot of time. But again, this premise is similar to a lot of chronic diseases. So my lab largely studies cancer processes and epigenetic dysregulation with cancer, but with aging processes as well, there's increasing evidence that suggests that epigenetic alterations are also a contributing factor to the aging process. So both DNA methylation, so this is a review paper that talks largely about how methylation alterations are affected during age, and in general, similar to methyl deficiencies, you see a global hypomethylation with age, but site-specific hypermethylation, so you can have both activation and gene silencing through the aging process. In addition, there is increasing evidence in not only DNA methylation, but other epigenetic marks, such as the chromatin-altering marks like histones, acetylation. So there's been already some discussion of these sirtuins in terms of the NAD-specific histone deacetylases, but even the other class 1 and class 2 HDAC enzymes also appear to be altered. And in addition, histone methylation appears to play a role. And I know, again, this afternoon we'll talk about microRNAs as well. Bottom line is, with age, there appears to be what's called, some people refer to as an epigenetic drift, where as we age, there is movement in terms of many of these epigenetic marks that influence gene expression that can contribute either to the aging process itself or to pathologies that are associated with aging. In addition to that, to add another level of complexity to this whole formula is that inflammatory processes also appear to contribute to altering epigenetic processes as well. And this may be mediated through direct cytokines. It may be mediated through oxidant stress. But again, there's increasing evidence that things like nitric oxide, I don't think my point, oh, here we go, and cytokines also influence enzymes such as DNA methyltransferases, histone methyltransferases to, again, change the epigenetic landscape, to alter, for example, chromatin access to TNF-alpha and IL-1 beta that exaggerate the pro-inflammatory response. So it's all becoming much more complicated in terms of trying to understand how all these processes are interrelated. So we've shown in our lab a little bit of evidence in terms of how methylation status, for example, affects inflammatory processes. So this is an in vitro study just simply using monocytic cells, so THP1 cells, where we stimulate them or try to activate an immune response with TNF-alpha, with LPS, and we're quantifying TNF-alpha and IL-1 beta response. So here in untreated cells, we don't see any big response, and what we've done is added 5-azacitidine, which is a well-known DNA methyltransferase inhibitor, so altering methylation status, and you can see there's a marked increase in pro-inflammatory cytokine expression when you alter the methylation status, clearly showing that changing methylation status makes a big impact in terms of chronic inflammation. We also know that zinc also plays a big role potentially in this process as well. So again, zinc's a part of a lot of different enzymes, including the DNA methyltransferases that control methylation status, these histone deacetylases, especially the class 1, class 2, within the active pocket is a zinc moiety. The methyl binding proteins that coordinate these two marks in terms of coordinating HDAC recruitment to methyl binding sites also is a zinc-containing protein. So there, again, there's been a lot of work trying to understand how zinc affects these processes. In addition, getting back to the methyl cycle, so those same enzymes that Dr. Zeisel talked about in terms of restoring acetyl methionine levels either through betaine or homocysteine, both of these enzymes are also zinc dependent. And it's clear from other studies that if you become zinc deficient, you also affect methyl status. But the effects of zinc on epigenetic processes is still relatively unstudied. So we can show when we take zinc deficient cells, so this is just culturing THP1 cells, that we can alter methylation status. So this is looking at global hypomethylation. Here we have 5-azacitidine as our positive control, and then as we make the cells zinc deficient, we see a change in zinc in hypomethylation status. I'm going to go through here a little bit more quickly. So I want to go over some of the data that we show in terms of an aging population. So here we have effects of just zinc deficiency. So using an in vitro model, and we can show again that when a cell is exposed to LPS, we see a rapid drop in intracellular zinc levels. And when they become deficient, the inflammatory response is exaggerated, where the zinc deficient cells have an exaggerated or increased TNF and IL-1 beta levels. So the zinc deficiency appears to be increasing the chronic inflammation. What happens now in the older animal? So again, reminding the older animal looks like a zinc deficient animal, even though they're eating adequate zinc levels. So why is this? So zinc transport, or intracellular zinc levels, are controlled by a whole family of zinc transporters. There's 24 of them. Some are influx, some are efflux. Their molecular regulation is not quite known. There's a lot of tissue specificity or cellular specificity in terms of their expression. What we did in the aging animals screen for all 24 transporters, and what we found was ZIP6 in particular was dysregulated in these aged animals. And interesting, ZIP6 is one of the key transporters that is expressed in immune cells, especially dendritic cells, and are very important in terms of their maturation and their antigen presenting and pro-inflammatory state. And so here this is the data. So here are the animals at increasing ages. Alone, we do not see a big change in ZIP6 expression, but when exposed to LPS, we see in the young animal a big increase in ZIP6. that we do not see in the older animals. And this is seen in other models as well, where we think that the ZIP6 is used to try to import more zinc to help protect the cell from insults. And in the aging animal, this response appears to be modified. Similarly, when we look at DNA methylation in the older animal, globally in the older animal, similar to what other people have seen, we see a big decrease in global methylation, but we see site-specific hypermethylation of the ZIP6 promoter. So there's a specific hypermethylation of ZIP6 in these older animals that may be accounting for that increase or decrease in cellular zinc levels and accounting for their increased susceptibility to zinc deficiency. And then to just show we've also knocked down ZIP6 in these primary cell lines as well. And we can see similar decreases in zinc and also exaggerated immune response, clearly showing that ZIP6 plays a role in exaggerating this immune response. We also see in these animals increased inflammatory processes. So here's IL-1 beta, and also in response to LPS. So basally, we see increased inflammation. And then with zinc deficiency and a stress, this response is markedly exaggerated. And then the take home is, so what can we do with these older animals? Can we supplement them with zinc to help alleviate some of these problems? And the answer is yes. So here we have zinc supplementation. So we supplement these animals with about 10x. So the young animals are getting, the controls are getting about 50 parts per million, and then the older animals are getting about 300 parts per million, and you can see that the older animals have the lower zinc levels, and the zinc supplementation is bringing them back to control levels of the young. And our bottom line is here, supplements really do protect against the age-related chronic inflammation. So the zinc supplements, both looking at IL-1 beta and IL-6 levels, appear to restore the chronic inflammation in these animals. So our bottom line is try to get some zinc, especially if you're a little bit older. So I won't go over that data. I've already explained some of it. But bottom line is, again, we don't really know where this cycle starts. So it's kind of chicken before the egg. Is it the age, the insult? Is it inflammation that's an insult? Is the loss in zinc the primary thing? We don't really know. But we think all of these things feed forward together to cause chronic inflammation, and that together, by improving zinc status in an older individual, that may help prevent against some of these defects. And our RDAs currently do not adjust for age aside from very young. If you're an adult, 18 and above, the requirement for zinc is the same. And we think there's a lot of data that suggests that maybe age-specific requirements for zinc, and zinc may be an age-essential nutrient that we do need to ensure we get enough, especially as we get older. So this is finally I just wanted to show the lab, but that's all I have. So I'm happy to take some questions. I know I'm holding you from your wine and your lunch, so. Who's asking the first question?
Thank you for the great reminder to watch our zinc levels. On a practical level, what is your preferred delivery of zinc in terms of bioavailability? It's probably not a zinc sulfate, it's probably a protein-bound compound. What works best?
Well, I'm very much a proponent of food-based first, so try to make sure you consume protein-rich foods. In terms of the bioavailability issue, so zinc sulfate, zinc gluconates are the most commonly available supplements. The protein-bound ones do have better bioavailability, but the increase in bioavailability is not a huge amount. So even consuming, so if you can't get enough zinc through your diet, consuming a zinc sulfate supplement at the 11 to 15, usually they're about 15 milligrams, should be adequate.
So you showed that with the zinc deficiency that the plasma level of 0.8 milligrams per ml does not change. What about the aged serum? Do the levels go down?
With age, yes, that we do see. There's quite a bit of variability. So if you look at other people that have done this work.
Do you attribute that to
It's a great question.
Location and zinc fingers, you mentioned zinc fingers.
Yes, and we don't know. We have attempted to profile all the zinc transporters in some our human studies and stratify to age, and our bottom line at this point is it's highly variable. So there isn't a single transporter that seems to pop up in terms of a potential candidate. So I think the story is much more complex, but we're still looking for sure.
Nice talking to you. Zinc levels affect, in great extent, the levels of metallocyanine, which is a very potent antioxidant.
Correct.
So, some of the effects of zinc deficiency could be ascribed to impairment of metallocyanine levels, and zinc supplementation, some of the beneficial effects of zinc supplementation could be ascribed also to an increase in levels of metallocyanine.
Yes, that's a good point. To be honest, though, in some of these marginal zinc deficiencies, the metallothionein levels don't appear to alter that much, or they're very acute, and then even over a long term, you don't see, even though we see dose-dependent, I guess, increases as we go longer in terms of inflammatory processes, we don't see changes in metallothionein that further change. So I think in terms of the early effects, metallothionein is playing a big role, but as you are more chronically deficient, there's something else that's going on, I believe.
Thank you.
The FDA has approved zinc L-monothionate in animal husbandry, veterinary, as an animal feed.
Right.
So since that is the FDA recognized form, do you have any comment on that? Zinc L-monothionate.
I don't know. So as you mentioned, it is used in a lot of feeds. I work quite a bit with some animal production companies that are trying to investigate bioavailability of different chelates, and the bioavailability does appear to be enhanced in those in these chelated protein forms. There just isn't enough data. No one has really used them in human models yet, though, in terms of trying to understand. And I think the need. Again, zinc sulfates, even though they have a decreased bioavailability, they do appear to change zinc status, so there doesn't appear to be a need to try to do alternatives at this point.
So we will end the session. Thank you for all. And